and welcome to Teach Me in 10. In this discussion, Dr. Jean Dunn shares valuable insights gleaned from her research measuring cytokines to assess cellular responses in clinical and research settings. And we'll be looking at unlocking cellular response with simple Plex technology. So without further ado, let's get into the episode. Hi, Jean. Welcome to Teach Me in 10. So, as we only have 10 minutes, we best get started. And my first question for you has to be can you briefly outline your research's focus, understanding cellular response and its implications for disease modifying drugs?
Hello, Lucy. Yes, absolutely. So, we're interested in the cellular response in common viruses and how they relate to diseases such as multiple sclerosis, which is a research focus of ours. So, currently, research into viral association in multiple sclerosis has focused on the Epstein Barr virus, or EBV. And this is a very common virus which most people easily deal with in childhood or adolescence. It's called infectious mononucleosis or kissing disease. Okay, so what happens is that the virus is controlled by immune cells which patrol and deal with any exposures. And a very large studies, I think there were 10 million samples included, uh, from US military personnel, identified new adult infection with Epstein Barr EBV as a prerequisite for subsequent development of MS. So that links MS causally with Epstein Barr disease. And that study measured antibody responses. Now, we've carried out a subsequent study that's been happily published this year in 2024, and our study looked at the cellular responses to Epstein-Barr in patients with MS. So we looked at various therapies that are used to treat patients with MS, and we looked at the impact of these therapies on the cytokine responses of immune cells that respond to Epstein-Barr. The piece of Epstein-Barr that we focused on it's called EBNA1, or Epstein-Barr nuclear associated antigen 1.
Perfect. So then from that, how have you developed the in-house cytokine release immunoassays and how are these aiding clinical researchers in studying inflammatory cytokine response?
So we base the assays on tests that we routinely use for latent TB. It's called the interferon gamma release assay. And these assays are commercially supplied in a kit with TB antigens. Blood is added, patient blood, it's incubated overnight, and an ELISA is used to measure interferon gamma. So the in-house assays that we develop use viral peptides or whole recombinant antigens. Now, they can be from any source, but the ones obviously that we used in the MS studies are EBNA1 from Epstein-Barr. So these are added to whole blood and cultured overnight, and then the cytokines produced by the blood cells are then measured in the supernatant. And this is a very flexible assay system, and Ella provides the great advantage of multiplex cytokine measurement on a single sample.
I see. So, what were the main challenges in creating these diagnostic tests, and what kind of key lessons were learned?
Well, for us, the main challenge is optimizing the assay. So that means establishing the amount of antigen that's required to elicit a response. And then after that, deciding on the appropriate cytokines to measure. So it's important to be able to identify a responder group to establish the assay, because you have to have a positive and perhaps a negative control. And these can be healthy controls, or they may be part of your disease cohort, as in the MS study. So a key lesson for us was to conduct a pilot study, including reasonable numbers of healthy controls, and within the patient cohort to identify subgroups. For example, in the MS study, we didn't initially include treatment naive patients because they were fewer in numbers. But eventually, this cohort proved to be essential for comparison. They were our baseline comparator. So, another key lesson is to identify the cytokine signature of the cellular response you wish to measure. For T cell responses, we used interferon gamma and IL-2 eventually. But when we conducted pilot studies, we included a number of other T cell associated and innate associated cytokines. And in an ideal world, a broad range of cytokines would be measured, but cost is a limiting factor, I'm afraid, in reality.
Absolutely. So, what would you say are the essential capabilities you seek in immunoassay platforms for diagnostic monitoring?
Well, reproducibility is essential. We need a low coefficient of variation for inter and intra-assay comparisons because we need to be able to rely on the results over a period of testing. Um, a wide dynamic range is a great advantage, and a wide range of sample types, such as plasma, serum, stimulation supernatants, and perhaps cerebrospinal fluid. Also, for our purposes, a wide range of cytokines, chemokines, or other biomarkers to be available for testing on that platform because that gives us a great possibility in what we look for. Ease of use with minimal requirement for operator intervention during testing is obviously a great advantage, and short setup times and short daily and monthly maintenance. Another capability would be low volume of samples because these are often precious samples, and quick time from initial sample testing to results generation because we're anxious to get the results, and obviously good engineering support and training.
Absolutely. And then from that, how do patients benefit from using the Simple Plex platform on the Ella instrument for cytokine measurement?
Well, all care is patient centered and it should be personalized. And on the Ella platform, the ease, reproducibility, and speed of measurement of the cytokines makes it possible to upscale and translate these assays, which are essentially a research assay, but then they need to be translated into use in the clinical laboratory. So the Ella platform has enabled us to develop novel assays to measure these individual responses. We've been able to look at various cytokine responses and a dynamic system, allowing us to choose the cytokines that best represent the cellular processes we want to measure. And the initial studies paved the way for things like longitudinal studies, looking at response to treatment, and possibly predicting changes required in dosage or treatment type.
Nice. So, what are the strengths and weaknesses of the Ella instrument, particularly regarding its capacity for measuring multiple cytokines on limited samples and its high sensitivity and reproducibility?
Well, as you say, there are great advantages in being able to measure a range of cytokines on a small, precious sample. And this is especially true when developing and optimizing assays that we will translate to the diagnostic and monitoring service. So a single step test for a range of cytokines saves time and human resources, and it produces a huge amount of information that we can then analyze to identify the most useful measures, so the most appropriate cytokines in our MS study. So the sensitivity and reproducibility are also essential in order to be able to trust results. You need to be able to trust that if you test it today and if you test it in 10 weeks, it will be the same. And also, we need to trust them because these results will change as we do longitudinal studies in each patient. It may be change in response to disease development or in response to treatment. A disadvantage is the in-house assay should be translatable into an IVD or CE marked product to enable us to use it in the clinical diagnostic setting. And this is something that needs investment of both time and resources.
I see. So, I'd love if you could share your experience in creating a novel cytokine release assay and the significance of using a platform that offers this fast, hands free operation.
Absolutely. So, most people with multiple sclerosis have high antibodies to a latent particle of EBV called EBNA1. And as I said, previous studies have used this antibody to demonstrate Epstein Barr reactivation. However, when we looked at these antibodies, we show that they didn't change in response to therapies that are used for MS. So, antibody levels can't be used to monitor treatment responses in patients. In contrast, when we developed the cellular assays and we looked at the cellular responses to EBNA1 using interferon gamma and IL-2 multiplexes on the ELA, we showed that the majority of new patients had a cellular response to EBNA1. And these are the treatment naive cohort, so they've never been treated. And this response, this cytokine response to EBNA1, was significantly decreased in patients treated with immunosuppressive and anti-B cell therapies. And these therapies mostly work by suppressing the inflammatory response or by eliminating immune cells. So there are two types of treatment. And a different therapy, natalizumab or Tysabri, works by preventing the cells from crossing the blood-brain barrier. Okay, so in that case, all of the MS patients were shown to have a high interferon gamma and IL-2 response to EBNA-1. And this indicated that the cells were held in the peripheral compartment, so they're held in the bloodstream, and they don't cross over the blood-brain barrier to create damage in the central nervous system. And these are all actually really novel findings that are enabled by the use of the ELA platform. So we were able to measure a range of cytokines in the pilot study. The supernatants were small, around 200 microliters. So the panel design capability in multiplexing, together with the range of available cytokines, was really useful for the initial pilot. And the results identified the relevant cytokines that we went on to test in the full cohort. So the short preparation time and the reliability and the speed of results for us that makes us very happy to get quick results. And it makes the ELA a great platform, most especially for the multiplex parameters. In general, there were very few assay failures, and there was very low inter and intra-assay coefficient of variability. In our hands, less than 2% for interferon gamma and IL-2, which
Wow,
is great. Yeah, it is very impressive.
incredible. And then finally, as we're almost out of time, what potential long term research advantages come with employing fast and efficient analytical techniques like this?
Well, the great advantage of the multiplex is that we can quickly and reproducibly, and that's important, test a range of biomarkers, including cytokines and chemokines, on these precious primary samples or in supernatants. So, as I said, we've used these cellular assays in the multiple sclerosis study where we looked at EBNA-1 and the changes following treatments. But this work will lead on to longitudinal studies where we've followed patients through disease flares and treatment changes. And the multiplex capability of ELA will allow us to test large numbers of samples easily and quickly. We only use 5 mils of blood, and this is much easier to take compared to a cerebrospinal fluid, which requires a lumbar puncture and an in-hospital procedure. And that's the current diagnostic biomarker in multiple sclerosis. So in other studies, to expand it, we've used SARS-CoV-2 peptides and antigens to measure cellular responses pre and post-vaccination in patients that are immunosuppressed. So these include patients on immunosuppressive therapies and those with inborn errors of immunity. So babies that are born, with defects in their immune system. And these are an important group because they may need individualized vaccination regimens to ensure protection. They won't always be protected by the same regime as other cohorts. And we're currently extending the studies from the SARS-CoV-2 vaccination to look at other common vaccine responses in immunosuppressed patients. So that's been the potential long-term research advantages for us.
Perfect. Well, thank you so much for joining us here in this episode. It's been absolutely wonderful speaking with you.
Well, thank you. It's been lovely to have your support and care.
Thanks so much for joining us in this episode because this episode has been brought to you by Biotechne. Biotechne is a global developer, manufacturer, and supplier of high quality reagents, analytical instruments, and precision diagnostics. Whether you're at the cutting edge of academic research, translating basic discoveries to therapeutic leads, or at a facility that requires the highest level of diagnostic testing, their award winning tools and solutions empower scientists and clinicians to achieve reproducible and consistent results. Trusting Biotechne means choosing confidence that every solution you use will help move you towards better answers. We've left some further resources below should you like to learn more, but thanks again for watching this episode, and I hope we'll see you again very soon.